The overall goal of the following experiment is to analyze the motion of choroid plexus epithelial cells cilia ex vivo. This is achieved by first excising the brain from a mouse embryo or pup to isolate the choroid plexus tissue. The motile ciliary movement is visualized by differential interference contrast, or DIC microscopy, and then a digital movie of the ciliary tips is acquired for manual tracking of the ciliary tip positions. Ultimately, the reconstituted tip movements of the cilia are categorized and quantified based on the patterns of their motions.
This method can help answer key questions in cell biology, such as how is ciliary motility controlled during development, and how does pattern of motility change over time.
The implication of these techniques extend towards pathophysiological analysis and diagnosis of cilia-associated diseases, as our fundamental understandings of kinetic cilia is still sparse.
Begin by placing a 70% ethanol-rinsed neonatal mouse head in ice-cold Leibovitz L15 medium in a sterile 10-centimeter dish immediately after decapitation. Next, use a pair of watchmaker forceps to remove the scalp from the skull. Then, cut open the skull by median section along the longitudinal suture to expose the brain, followed by cutting of the cranial nerves to harvest the